Dr. Guru Prasanna, and the convener G.K. Sujayanand, Dr. G.K. Sujayanand, and the rapporteurs Dr. Suganya Kanna and Dr. Srinivasan. So now let us start the session with the lead talk by Dr. Arun K. Shankar.
Good afternoon, participants. It's my privilege to introduce the keynote speaker, Dr. Arun K. Shankar, Principal Scientist, Plant Physiology, working at CRIDA, Central Research Institute for Dryland Agriculture, Hyderabad, India. With over 30 years of experience, his research focuses on crop responses to abiotic stresses like drought, heat, salinity, and heavy metals, particularly in photosystem dynamics and stress tolerance mechanisms. Dr. Shankar has received international recognition, including the Endeavour Executive Fellowship, Australia, and has been listed among top 2% of the world scientists. He has published extensively with 7,000 plus citations and an H index of 31, and serves as an editor and reviewer for leading journals such as Scientific Reports and Frontiers in Plant Science. His expertise in bioinformatics and protein modeling has advanced climate resilient agriculture, and his collaborations with institutes like CSIRO Australia and Virginia Bioinformatics Institute have significantly contributed to high resolution phenotyping and crop stress adaptations. We are honored to have Dr. Shankar with us today. Sir, please.
Thank you very much, Chairman and Co-Chair. Firstly, I want to thank the organizers for having invited me for this lead talk in this session of nutrition and physiological aspects for integrated pest management. Actually, thank you for the introduction. I actually work mainly on abiotic stress, and this is the first time I'm attending a conference in biotic stress, and my main work is on drought and heat and cold stress, heavy metal stress. So I have tried to interpolate my work on how it can be transposed into plant pathogen interactions. I work mainly on I concentrate mainly on photosystem 2 dynamics, that is photosystem 2 electron transport and chlorophyll fluorescence as a tool for detecting stress, and as it also mainly on the mechanism of how photosystem 2 is affected by either biotic or abiotic stress. My work basically is on abiotic stress, like I said, but here for this as a theme to get in tune with the theme here, I have concentrated a lead talk mainly of, I have put in through a lot of other works together and tried to set it up in such a way that it can help the audience here. So the title here is that chlorophyll fluorescence and photosystem 2 dynamics under plant pathogen interactions and tools for early detection and integrated pest management, disease and pest management. So the key overview of the talk will be on chlorophyll fluorescence as a non-invasive diagnostic tool for detecting stress, and links between photosystem 2 efficiency and pathogen-induced physiological changes when there is a disease infestation. Then the role of fluorescence data in integrated disease management, how we can use the fluorescence data in either detecting or in rapidly detecting, rapidly screening, and also controlling to an extent. And also future integration of artificial intelligence driven analytics and precision agriculture. So firstly, I want to show you the structure of the photosystem 2, because that is where the whole lead talk will be centered on. The top part is the the top part is the Z scheme, which most of you would have seen. The bottom is the enzyme, that is oxidoreductase enzyme, plastoquinone oxidoreductase enzyme. This is a molecular model of that, and more usually the three main components are photosystem 2, cytochrome B6, photosystem 1, and ATP synthase, which is the whole of the light reaction. And the concentration of my work here and my talk here will be on this part of it. That is photosystem 2 dynamics, just after water splitting and the electron which passes through QA to QB. This is where I'll totally concentrate on this. And this is where most of the diagnostic tools are centered on. The instrumentation now has sophisticated enough to detect electron transport here. So before that, I'll just briefly tell you what actually happens when the photon strikes. That is, water splits and releases 4 electrons into the which when the photon strikes the chlorophyll atom, it goes to excited states and electron transport starts, and it is replenished by water splitting. So the oxygen we get here, the whole of the oxygen in the earth as such, whatever oxygen we have in the atmosphere is because of this reaction. This is the only sole supplier of oxygen to all of the planet of Earth. So if we have oxygen on Earth, this is because of this water splitting. And now what I do is I measure the electron transport here. The electron transport starting from the chlorophyll atom to reaction center P680 to QA to QB. I don't go up to cytochrome complex, QA to QB. And this is where I measure the electron transport and I extrapolate it in how we can detect and manage diseases. Or I have worked on abiotic. Now I'll go into extrapolate onto how we can go into abiotic stresses. This is the molecular model again. So this is called the Kotsky curve. That is it is named after the discoverer of this curve. This is actually the OHP transient. That is it is fast fluorescence kinetics. That is what we measure with our instruments. Fast fluorescence kinetics is measured. That is when the photon strikes the chlorophyll atom. There are 3 types of things that takes place. One is that fluorescence, one is heat, and the third is the photochemistry. Photochemistry is the process by which we get our food. That is main process by which chemical energy is converted into food production. That is CHO is produced, and the wasted energy is fluorescence and thermal dissipation. That is non-radiative dissipation. So this actually measures that, and we can correlate as to how much of photochemistry is taking place and how much is wasted. And that is how we go about trying to find out or detect either a biotic stress or abiotic stress. So this curve is this is a typical fluorescence curve which takes place from 10 to the power of -6 seconds to 10 to the power of 4 seconds, and it is split into 4 parts. That is, O is this region which represents when all the reaction centers are open with no elimination. Then this O and J. OJ is the region where photochemical reduction takes place, QA to QB, which I told you before. And J is the reduced state of all the reaction centers in the chlorophyll, QA, QB, Q2, and Q2. Then it goes to plasticmenon. And the last state is which is at 300 milliseconds. That coincides with the maximum concentration of QA and QB. So this I measure with my instrument. This I measure within the measure. I pulse light. I'll show you the instrument next slide. So the main area of focus will be. Oops, sorry. So the focus of the talk here, the focus I'll be doing on is the mechanistic insights, how photosystem 2 inhibition mechanisms during pathogen attack takes place. That is how our ROS accumulation takes place, how electron transport is disrupted. There is a chain, that is a traffic jam like situation takes place in the electron transport, where it is concentrated at a particular step, and that reflects on symptoms. And this is a biomarker identification. Actually, what are the parametric identifications we have to do in order to rapidly screen? That is FV by FM, which is a quantum yield. It is for efficiency of photochemistry. NPQ is the non-photochemical quenching. These two are the important parametric biometric biomarkers which can be used as early stress biomarkers. Then infection stage correlation, how we correlate these parameters with infection stages, that is linking it with the fluorescence pattern to infection progression. Then how we integrate data, combining this data with hyperspectral, which which gathers data in other wavelengths other than fluorescence, that is a radio wavelength and higher wavelength in the electromagnetic spectrum, and how environmental sensors can be used, and further pathogen genomics. So then validation will be the next step, where we test across pathogen symptoms, and like for example, these are examples we can test across, and how really the ground truthing is actually correlates with our parametric assessment in fluorescence. So the workflow is actually, if you look at the workflow, first we have data acquisition. That is, we either take quantitative measures. That is, we measure in terms of numerical numbers like FV by FM or quantum yield. We have a plant which is optimum in optimum conditions. That is, if environmental conditions, it has a quantum efficiency of 0.7. 0.7 is that no system in the universe, or rather in the planet, has got 100% efficiency. We know that the maximum efficiency for any biological process is about 70%. So the QV FE by FM will be 0.7 for a healthy plant. That is 30% of the energy that is falling on it is wasted in terms of fluorescence and heat dissipation, and 70 is used for photochemical transfer for food production. So this particular data acquisition we use. That is, when it goes down below 7, we assume that there is a disruption in electron transport, and there is either a disease or there is a biotic or abiotic stress. Then extraction of the parameters, that is FE by FM, photochemistry, efficiency of photochemistry, and all the electron transport parameters, which is a very complex one, and we measure that within milliseconds. I'll show you the instrument and how to measure it also. And we detect dysfunction in the photosystem too. Then we do multi-model integration. This is actually the workflow. So we combine this data with the rest of the other data like soil, weather, soil moisture, pathogen load with biochemical work. Then finally we do AI predictive modeling. We can train models. We can use convoluted neural networks, forest randoms, and several other algorithms, classify signatures, and we can build a database, and we can actually transpose our symptoms on the database, and the AI will detect it and immediately tell us the severity of the disease, and also further it goes about to tell even the control measure. So this is the workflow. Actually, the slide is not fully shown. Decision support system is the final one. So this is the instrument I use. This is a chlorophyll fluorescence meter, which actually this is the sensor I place on the leaf. This can be a stressed leaf, either biotic or abiotic. And here we get the readout. And this sensor pulses actinic light. That is a photosynthetically active radiation. It pulses on the leaf surface, and actually I clip the leaf surface and dark adapt it for about half an hour. So when the leaf surface is dark adapted for half an hour, all the photosynthetic process stops in the leaf. That is, there is no photon falling on the leaf, so it comes to a standstill. What happens like in the night? So after that, after having dark adapted it for half an hour, I place the sensor and pulse actinic light. When I pulse the actinic light, immediately photosynthesis starts to take place, and photon strikes on the chlorophyll atom, and I showed you the biochemical train pathway. That is a Z scheme that starts to take place. That is, electron starts to move, and the sensor starts to measure the electron transport. It measures the electron transport, gives me numerical values in this. So this is how I measure it. This is the instrument, like this is a sensor, this is the clip, and these are the parameters I get. FV by FM. The FV by FM is the, like I told you, 0.729. You can see here. If you can see that is a healthy leaf. So if it goes below that, about 5 or so, it's a stressed leaf, and it won't go beyond 0.79, 729, 75 is the maximum it will go. And there are a whole gamut of parameters that we get here. And we can analyze, and we can deduce, and we can do a lot of equation mathematics to get a lot of other parameters from this. So this is the leaf clip. I told you how I dark adapt the leaves. For example, if this is a diseased leaf, I dark adapt the leaf with a leaf clip so that this particular area is dark adapted. That is, no light falls on it. Then afterwards, I place the sensor there. So now coming to what exactly happens in the leaf when disease is affected, and how I break it down into my fluorescence parameters is that first there is a chlorophyll breakdown, there is a general generational and scavenging of ROS taking place. Then this is the key step, that is inhibition of photosystem 2 photochemistry. When photochemistry is inhibited, there is no food production. That is the ATP and NADP are not synthesized and the dark reaction does not take place, or at least it is disrupted. Less dark reaction takes place, less food production takes place, and less yield. You can keep on jumping up to the final yield level. So the damage, oxidative damage, and this is heat shock protection. This is a protective area. Inhibition of Calvin cycle, like I told you, dark reaction is inhibited. Then degradation of proteins. There is a repair cycle also taking place by metabolic reprogramming. This is what happens. It is generally the same thing even in a abiotic stress also. Now I showed you about parametric estimation, numerical estimation of photosystem photosynthetic efficiency. This numerical efficiency, there is a drawback in that because I showed you the place where I actually measured. That is about 1 mm diameter area where I place the sensor and where my actinic light is pulsed. And I get numerical values 0.7. Now here this is the modern way where we do imaging. Because of because numerical values I can only take in a particular pinpoint. If the leaf area is about 10 square centimeters, my data acquisition is only in 1 millimeter squared. And when a disease is affected, disease affects the plant, it is not necessarily that particular 1 millimeter square will have a disease symptom. It can be there might not be any lesion there. Then it might be healthy part of the leaf also, and photosynthesis might be taking place actively there. So I might miss the parameter acquisition. So to overcome that, now the systems have changed over to imaging. We image the whole leaf. That is the plant is actinic light is pulsed in the whole leaf, kept in a dark chamber. And the image, we get the image. This image is the fluorescence image of the leaf. That the color is the fluorescence emitting back. When actually what is fluorescence? It is when your chlorophyll atoms comes down to from excited state to normal state, it dissipates longer wavelength energy, and that is imaged by this. So that by imaging we can actually analyze pixel by pixel of damage taking place. Like we can enlarge this image, we can pixelate it, and each pixel will know the numerical values. Like if I do with my instrument, I get only one 10 millimeter square area value, but here I get 1 pixel by pixel. I can get about 1 million data here, 1 million data points here. So by exposing the plants to a very bright flashlight, saturating light pulse, reaction centers are closed and saturated. Then resulting fluorescence can be measured. Images are processed in the cameras and extracting meaningful physiological phenotyping data from this. So then I can get a quantum yield of whole of the leaf. I can average it, and also I can find out whether, suppose the disease symptom is occurring here only, or even say the insect infestation symptom is occurring here only, then I can find out exactly how photosystem 2 is affected in that particular area of lesion formation. Now I'll show you some examples. This is one of the classical example where chlorophyll's fluorescence imaging was done in a barley infected B. graminis pathogen. It's a broad spectrum MLO, race specific. Here this is a control and this is a susceptible one, and this is the numerical data put graphical representation. You can see that the control has got a electron transport is quite normal and going on in a correct way, whereas in the susceptible and the disease affected plants there is a lot of deviation from the control. This is taken as a this can be used to predict disease progression much earlier than what visual RGB can predict. That is, for example, 3 days of infection in ATR regions and lesions adjacent to the leaf, and the remainder of the leaf can be easily taken by images, and we can predict the progression of the disease. This one again is there are some. First, I'll go back to the. There are actually algorithms that are introduced in the image analysis here. There are random frog, sequential forward selection, and Monte Carlo algorithms, which I don't want to go in detail, because that will go beyond the scope of the lecture. Although I'll give you in a sentence, I'll tell you what it is. It is an iterative variable selection algorithm that randomly searches for high dimensional data. And this is iterative, but it pinpointed searches, that greedy feature it is called. Then we have Monte Carlo is a very common one, and it filters out the data. So these algorithms are used in the image analysis here. And we can even differentiate between nutrient deficiency and also HLB, that is HLB is a disease. Hulong being disease of citrus, which is a quite devastating disease in North America. So this disease infected and nutrient infected, and we can see that how these three algorithms differentiate. That you put to use all the three algorithms and find out the progression of the disease, and we can see actually what is the difference. And this is fed into your database, and the AI algorithm captures this, so that you have several hundreds of thousands of images like this. So when you generate hundreds of thousands of images in these three spectrums, that is all the three algorithms, this can be used to immediately predict your disease and immediately differentiate between a disease infected and a nutrient deficient symptom. All these are, that is a Fe by Fm. Like I told you, this is Fe by Fm is a quantum yield, and this is a non-photochemical quenching. This is Fe by F0, that is initial fluorescence. These are parametric, that electron transport parameters, which this imaging measures. So from this we will be able to quantify, and also we'll be able to predict the progression of the disease. This one I showed you. Now this again is another type of imaging. This is another example within say from 2 to 300 milliseconds. Like I pulse the light, I get my data from 2 to 300 milliseconds in my instrument, and you can see the progression here, how a control and a disease affected progresses within, like the parametric acquisition is within the 300 seconds. And in 300 seconds, you can see the difference between control and the disease affected ones. And I can again graphically represent the data here, and I can pinpoint pixels. And this way I can pathogen induced damage to photosystem 2 can be quantified, either viral, bacterial, or I can extrapolate. This is an example here, but I can do that for viral, bacterial, fungal, amylo induced, anything, because all are going to affect the photosystem 2 anyway. And mechanism is, like I showed you in previous slides, ROS accumulation, light blockage of light interception, blockage of electron transport, that is causing a traffic jam in electron transport, metabolite theft. That is because the disease causing organism is also having its own energy synthesis mechanisms, and in all probability it can even have chlorophyll. So it does metabolite theft, and senescence acceleration also takes place. So now again another example I'll show you of mosaic virus. That is virus tagged in these are the days after days post inoculation of the virus, and this is in green fluorescence protein. This is the normal RGB image, and this is a fluorescence image, which this is the NPQ image. So here virus tagged with green fluorescence protein, it glows. You can see green fluorescence protein is a very common usage in biotechnology. You know that it glows. That is one method. And now this is natural color. The RGB is natural color. And here we get photosystem 2 efficiency measured 60 seconds after light exposure. And just you can see the difference between how 6 days post inoculation, and here it is 10 days post inoculation. That is 4 days of disease progression. So both your photosystem 2 efficiency and non-photochemical quenching, these two are inversely proportional to each other. That is, if you have a damage in photosystem 2 efficiency, then your non-photochemical quenching increases. That is the reason you get the damage. That is, more of the energy is dissipated as thermal heat. So this way also we can predict, or we can actually assess the damage caused by pathogens on photosystem 2. Here is the dynamics. Like here you can see the dynamics of chlorophyll fluorescence parameters during the progression of disease caused by various pathogens. That is a virus, bacteria, and fungus. And the color intensity of each graph reflects on the degree of impact. The dotted line shows the control value and parameters. You can see the difference between the control and disease infected plants, and also the parametric differences like NPQ, quantum yield, and efficiency of photochemistry. Now, future perspectives of this is that this is a very emerging area. That is, not much of work has been done in this. In fact, I have also not started to work. When I got this idea, I'm thinking of starting working on this. And it is an emerging area, and it is not very, there is a very large potential for discovery. If anybody is like interested in this, and if you have the equipment for like, if you have that, the equipment I showed you, the Hansatek equipment or a Walls equipment, they are quite sophisticated and expensive, costly equipments. If you have them, you can get along, you can start off these projects here for students especially. And there is a great potential of discovery. That is, novel discoveries are possible in this. And the perspectives are the real-time diagnostics, AI models for instant fluorescence data, instantaneous data, then interpretation, and also predictive epidemiology, forecasting pathogen spread using fluorescence trends and climate data, then IoT integration, autonomous monitoring via AI-powered sensors, then resilience engineering, everything is AI guided, of course. Then I connect this with molecular biology and biotechnology here. Then collaborative platforms, federated learning, then ethics, of course, there is ethics involved in this. Whenever you use AI, you have to be very careful to be ethically correct. Then the conclusion here is that the key area of the talk was integrating fluorescence data, that is parametric data acquired by fluorescence imaging or fluorescence detection in integrated disease and pest management. So early disease detection, it can detect pathogen-induced stress before visible symptom appears. This is one of the important things that I'm going to stress in this talk. Before your visible symptom appears, it can be detected. If you are able to, and there are LiDAR systems. There are systems where you can deploy fluorescence imaging system above about 10 meters, and it images in a fisheye lens over large area. So you can cover about hectares of plant population in one imaging system, and you can have a screen which will show images, by which you can detect pathogen-induced stress much before visible symptoms starts to appear. Then you can differentiate. You can differentiate between biotic and abiotic stress, because there are specific fluorescence signatures that are specific to biotic stresses. And in biotic stresses, drought stress, heat stress, and specifically in abiotic stress, it also there are special signatures, and more specifically in biotic stress. That is, a fungal pathogen will have a specific signature, and bacterial pathogen and viral pathogen will have separate signatures. So you can differentiate between the organism that is affecting the plant much before visual symptoms appear. Then monitoring treatment efficiency, precision application, and reducing chemical use. That is, of course, the ultimate goal for us. Here I'll show you one of the imaging systems I used. Video ஓடுமாங்க எஸ்கேப் பண்ணிட்டு பாருங்க, கிளிக் பண்ணுங்க. இல்லையா? Actually, this is a video how I extract the video. பாருங்க என்ன பவர் பாயிண்ட் இது இல்ல வீடியோ இருந்ததுன்னா அந்த இது வரும், அது வரல, ஐகான் இல்ல. This is actually this is a automated imaging system. This trays, they go into a dark adapted setup. அதை கிளிக் பண்ணா என்ன அது பாருங்க, கிளிக் அந்த நத்திங்கா. So what happens is it gets inside a system, a closed system, a dark system, where light pulses on all the plants, and the image is reflected on your computer screen. This is a video, but unfortunately I'm not able to show it here. That's all. No? Thank you very much. Any questions you can ask, please.
Thank you, sir. Thank you for sparing your valuable time and knowledge with our participants. It was a wonderful session. So it has thrown a light to initiate such type of work in the near future. Actually, this type of studies will be very much useful in viral disease management, because early detection is very much important, so that we can save the crop also, sir, and further spread also. I think this technique may not be suitable for insect.
Insect.
Yes, sir. Yes, sir.
That's why I said plant pathogen interaction.
Yes,
only.
sir. Especially
The
for
type is
viral
wrong.
disease and MLO, it will be very much useful. So now the session is open for discussion. Students, you can interact with sir.
All of you are entomologists here, I guess. I've been put in the wrong place.
Actually, it's a very good study, sir, for especially in virus vector relationship studies and all. And for early detection, it's a wonderful tool.
But you can even think of making use of it in some insect related.
Yes.
That will be the novelty. In fact, if you are able to develop a system where you can predict insect damage much before visual symptoms occur.
Yes. Sir.
Sir, in one slide you had mentioned about we can detect reactive oxygen species. So I think in that way we can correlate with the insects. Some sucking insects, especially pod bugs or jacids, whiteflies, they also when they feed the reactive oxygen species in the plants will increase. So in that way, but further studies has to be validated whether sucking can be damage can be sensed through this photosystem fluorescence or not.
Actually, there is a catch in that. The reactive oxygen is more of a biochemical process than this biophysical process which I'm describing. So my instrumentation, which I work on, is more of instantaneous values when I put it with a sensor and get values of all the photosystem 2 electron transport parameters. But one of the side or one of the causes, one of the next steps is the reactive oxygen species. So that is definitely a biochemical step. Any insect damage or any stress is going to cause reactive oxygen species increase. All the three enzymes, that is SOD, catalase, and peroxidase, are going to increase. That is a normal standard procedure you have to do for any biotic stress or I suppose. Do you people in insect studies actually estimate reactive oxygen species in terms of enzyme activity? Do you actually do that? Do students do it here? I'm asking you people. Not much of biochemistry, I don't think you do. No? But that will be novelty, that you for if you look at if you want to publish in very high impact journals, this sort of data will add to your.
Sir, thank you for your excellent presentation, sir. I have a small query. As you were telling that the fluorescence-based detection, as earlier also we have that hyperspectral remote sensing related detection. That is also in the down the line around 10 to 15 years they have detected and find out the different wavelength. That is also giving the advantage only for the window period of around few days. That if the days are very much less, that because even though we can go for the control measures, then we cannot avoid the damage. So what is the, can you throw your light, some light compared to that hyperspectral image sensing, how fluorescence maybe can solve the problem of giving that time advantage?
Fluorescence, hyperspectral, definitely fluorescence can advance your time by 3 or 4 days. Hyperspectral, now actually in my institute, Dr. Prabhakar is doing hyperspectral imaging in pest forecasting. That is NICRA PI Prabhakar, you must be knowing. He is doing a lot of work on that. But his window and his ground truthing, he is not able to predict diseases so much in advance as what LiDAR related fluorescence can do. But I don't know if anybody is really doing that experiment. I have got my stuff from literature. I have not done it myself. That is why I said in the beginning that I am working on abiotic and not biotic. But definitely this is an advancement over hyperspectral, because this is dealing, hyperspectral is dealing with your atmospheric electromagnetic radiation window. Here we are dealing directly with what is falling on the chlorophyll, and because your pathogen is affecting your chlorophyll photosystem too, the effect will be much faster. And definitely you can do. My feeling is that definitely it will advance your detection period by a few days, and that will be a big advantage for control measure. But I don't know. I am not an expert in plant protection. I don't know when you people will take control measure, because before detecting you cannot take control measure. Before symptom comes you cannot take control measure. Threshold level, all those things are there. But this is just an idea that is I'm trying to tell.
At the same, sir, even the hyperspectral remote sensing, the wavelengths for insect pest, similarly disease, even the nutrient damage, it is similarly, we cannot segregate which much differences. It will be always fall in the, for example, saying that 450 to 470 nanometer, the other nutrient deficiency also coming between 480 to 520. So it is always nearer. Compared to that, in the fluorescence method, can we go for example, in the field condition, we are not going to detect disease or insect pest or whatever the stresses separately. There we are going to experience in the common platform. It may disease or it may be the pest, diseases or pest together or alone.
Yes.
In that conditions, this fluorescence method can able to differentiate. I don't know because work is not going.
It may not be able to. It may not, because if we have multiple stresses, you cannot tell what is the reason for the stress. I don't think it is possible. And
yeah, because
and in a field condition, there is always a possibility that there will be multiple stress. You can't expect only one
single
one stress. There will be drought. There can also be a disease. In all probability, when there is drought, there will be a disease or something. And I don't think in a normal farmer's field condition, you'll have only one stress going on. You'll have multiple stress. So the experimentally, like control laboratory studies, this you can mechanistic insight you can gain. What is the mechanism? See, more of a physiologist's viewpoint I'm talking. I'm talking about what is happening inside the plant.
Yeah.
Like we observe, like for example, you see, if you don't water the plant, it is wilting. So what a physiologist will tell is what is happening when it is wilting. What are the things, functional and structural aspects, how it is happening. Like question to what is how it is happening. That is what I'm trying to convey here. How the things are happening, what is actually taking place, how your electron transport is affected by disease or something. But multiple stresses will have the same method. Both your virus or fungus and heat is going to affect the photosystem too in the same way. So signatures, I don't know, maybe more AI has to come into this to get signatures, specific signatures.
Even it's detecting that particular thing, it may be a disease or insect, it will be good.
Some stress we can identify.
Yeah. Thank you, sir.
As a token of gratitude, I request Dr. P. Chandramani, ma'am, Professor and Head Entomology, AC and RA Madurai, to honor Dr. Arun K. Shankar, Principal Scientist, sir, with a memento.
So now we'll start the oral presentations. There are about 10 presentations. First, let me call Dr. K. Sengutwan. The second one, Dr. S. Jairani, ma'am. Okay. So the third is my presentation.
You can start your presentation, ma'am. So Dr. Suganti, she will be presenting on the topic harnessing asters for eco-friendly pest management in organic vegetable production. Ma'am, over to you.
Thank you. So today I'm going to talk about my research on harnessing asters for eco-friendly pest management in vegetables. So why this astras? Where it comes? So actually we all know the ill effects of chemical, that is indiscriminate use of chemical pesticides. The three are residue and resurgence, and apart from that, the indiscriminate use of insecticides, it destroys the natural enemies, as well as it cause serious ill effects or health issues in human being as well as in animal. So the best alternative is eco-friendly pest management, and the government of state government as well as central government, they are promoting this organic farming and natural farming, in which we promote this farm made concoction, especially in natural farming. We promote IPM, and for foliar spray we use this farm made concoctions. They are nothing but the astras. We are going to talk about those astras. So actually there is a myth. The farmers, especially the organic farmers and natural farmers, they have a myth that desi cow is important to have this or to practice this organic farming or natural farming. So we want to scientifically validate whether this desi cows are needed, whether it should be integrated as one of the components in this organic farming, or even the high milk yielding cows like crossbreeds can also be used. So the objective of the research is to study the efficacy of these astras made out of cow dung and cow urine, and this biocharacterization, as well as to study the safety of these astras on natural enemies. So the first objective is to study the efficacy of astras. We have used, actually there are 3 astras in insect pest management. The first one is neem astra, and the second one is brahmastra, third one is agni astra. So the brahmastra and agni astra, we prepare it by boiling, whereas this neem astra is prepared in cold water. It's a cold water preparation. So we have evaluated the efficacy of these astras in 3 vegetable crops, namely chili, brinjal, and tomato. And here we validated in such a way that we prepared 3 different formulations of these 3 astras. Neem astra had N1, N2, N3, and brahmastra and agni astra also had 3 different formulations. So N1 is nothing but N1 or B1 or A1. What does it mean? It is the astra prepared from the cow dung and cow urine of desi cow. Here we have taken Kangayam as the native breed, and the exotic breed we have taken is the HF, and that is the formulation number 2. And formulation 3 is the mix of native breed and the cross breed. So this is how we prepare. I don't want to waste my time on telling the preparation methods. We all know this. And these are all the treatments. So the first one is foliar application of neem astra. That is actually neem astra should be used without diluting with water, because already we are preparing in water. Some of the literatures, they are wrongly being published stating that neem astra is recommended at the rate of 10%. It is not so. It has to be sprayed as such without dilution. The second set of treatments, that is 4 to 6, are application of brahmastra 1, 2, 3 at the rate of 3%. That is 6 liters in 200 liters of water. And the 7, 8, 9 are foliar application of Agni Astra 1, 2, 3, the same concentration, that is 3%, otherwise 6 liters per acre. And we have taken this azadirachtin 10,000 ppm as a standard check and compared with this untreated control. So these are all the results. I'll go to the results. So this is the tablet. It's a pooled mean of 2-year data conducted during 2023 and 24. This is the efficacy of these astras on thrips and leaf curl caused by thrips as well as this mites and yield. So this T10 is the azadirachtin, T7 is Agni Astra 1, and T4 is the Brahmastra 1. I'll come to the results directly. So this is the data on the astras and efficacy of astras on shoot and fruit borer, because that was the only major pest in our experimental trial. And this is regarding the pest infestation in tomato. That is mainly we had only the infestation of thrips, this white flies, and leaf miner. So the results states that in chili, Agni Astra 1 and Brahmastra 1 at the rate of 3%, it was found to be statistically on par in the management of thrips and leaf curl damage. It was almost statistically on par with azadirachtin 10,000 ppm. And in the case of brinjal also, you see azadirachtin, Agni Astra and Brahmastra, these 3 were found to be statistically on par in the management of shoot and fruit borer. The infestation level, you see, it is only 3.9 to 4.3%. And in terms of yield also, this azadirachtin 10,000 ppm and Agni Astra, it was better. But in the case of chili, we couldn't find any yield difference in the, all the treatments. In the case of tomato, Agni Astra 1 and Brahmastra 1, it were found to be statistically on par with each other in the management of thrips, whereas Agni Astra 1 was found to be superior over Brahmastra in the management of white flies. With respect to leaf miner, all the 3 formulations of Agni Astra, it was found to be effective and it was statistically on par with each other, but there was no significant difference in the yield. The second objective is characterization. So we have taken up 3 types of characterization. One is biochemical characterization using GCMS and nutrient profiling of these 3 astras, and metagenomic characterization. So so though there were more than 50 different compounds in GCMS, I have just picked up only the compounds showing more than 5% area. So this, see, if you observe the compounds in the GCMS analysis, most of the compounds, they have either antifeedant or biopesticidal or antifungal, antibacterial, as well as they have some defense inducing, plant defense inducing molecules. So this is the biochemical characterization of Brahmastra, and this is regarding Agnyastra. So this is the microbial population. So this Nemastra are prepared out of this native breed cow dung and cow urine of this crossbreed and this mix of these both. Though they had the maximum population of bacterial colonies, fungi as well as actinomycetes. Since this Brahmastra and Agnyastra, they were prepared by, that's a hot preparation, we could get only the population of this bacteria, not the fungi and actinomycetes. This is the nutrient status. So with regard to Nemastra, the organic carbon content was maximum in the mix, that is Nemastra 3, whereas this N was maximum in N1, and this phosphorus was maximum in this Nemastra 3, and K in Nemastra 2. So it shows that, what does it mean? It shows that this cow dung and cow urine of Desi cow or crossbreed, it doesn't influence this nutrient status because it is not having much influence. When we talk about the astras, then we can say the efficacy not within the formulations. Then we coming to the metagenomic analysis. This is the Corona plot, which shows that this Nemastra and Agnyastra, they have wonderful microbial diversity. Especially the bacterial diversity was excellent in this Nemastra and Agnyastra when compared to Brahmastra. This is the relative abundance bar plot at the genus level for Nemastra. And this is, so actually this alpha diversity analysis indicates that this N2, that is the preparations from crossbreed, it has maximum bacterial diversity as well as the richness as well as diversity, it was maximum, as well as the evenness also, it is maximum in N2. This is the relative abundance bar plot of this Brahmastra. Here also you can see, but the abundance, the diversity, bacterial diversity is minimum in this case when compared to the earlier one. So in this case, in Brahmastra, if you see the bacterial diversity, this richness and evenness, everything is very good with respect to this native breed, the formulations prepared with the cow dung and cow urine of this native breed. And this is related to the Agnyastra. Here we could see, if you see the species level diversity, most of the beneficial organism, bacteria we could find in this Agnyastra. That is the advantage. See, most of the plant protection related biocontrol agents we could see in this, except E. coli. And this is the alpha diversity analysis graph, which shows that this B2 is effective with respect to diversity and richness, whereas this that is A1 is very good in with respect to this evenness of this bacterial diversity. Then this actually in all the three formulations, we had the major phylum of this proteobacteria, firmicutes and bacteroidales, which play significant role in contributing the soil fertility, plant growth and development, as well as plant protection. This Bacillus and Lactobacillus, these two were the major genus, and they stimulate the plant growth and induce the systemic resistance of the plants. From the chronoplot, it is evident that this Nemastra and Agnyastra, they have very rich biodiversity when compared to this Brahmastra. Then alpha diversity analysis indicates that this microbial diversity and evenness was maximum in Nemastra, prepared from the cow urine and cow dung collected from crossbreed, while it was maximum in Brahmastra and Agnyastra prepared from this native breed Kangayam. So the last objective is, it is with respect to the safety of this astras in unnatural enemies. So we have taken one predator, and the most frequently used parasitoid is the Trichogramma pretiosum in vegetable ecosystem, and the predator is the Chrysoperla jastrovisilami. So we tested these predator and parasitoid, the efficacy of these astras on these two natural enemies, and we found that this Nemastra at an undiluted form, it was highly safe to all these predators as well as parasitoids. So with respect to Trigogramma prettiosum, it is highly safe on adult emergence as well as parasitism. And with respect to Chrysoperla also, it is highly safe on the eggs, grapes, as well as on the adults of Chrysoperla. So when compared to Azadirachtin 10,000 ppm, these three formulations, these three astras, they were found to be highly safe on natural enemies. And among the three astras, this neem astra is found to be highly safe. The purpose of having these astras in natural farming, that is, it's a farm-made concoction. The farmer, they can prepare it themselves in their own farm with the available resources. They need not depend on the external inputs. The purpose is, actually, it will not completely kill the pest population. Some residual population will be there in the field on which this predators and parasitoid can work well. That is the reason now, since we are promoting IPM, along with these concoctions, we could able to achieve very good protection in the natural farming as well as in the organic fields when compared to the conventional farming. In my one of the poster session also, I'm presenting another paper, that is how we have compared different farming systems, namely organic, natural and conventional in pest management. So in that I have given elaborately. So when apart from monetary benefit, sometimes the monetary benefit may not be good in organic or natural farming, but in terms of ecosystem services, definitely it helps us to for sustainable agriculture, we need this type of farming system. Thank you. So if you have any doubt, you can ask.
The topic is open for discussion. Any questions?
Okay.
So with the time shortage, ma'am,
Okay,
we will
thank
take
you.
the question later.
Thank you for the opportunity.
Thank you. Thank you for your questions.
So now I call upon Dr. Suganya Kanna.
Good evening to one and all. And I'm here to present my paper on the impact of field level demonstration on IPDM for the management of pest and disease in Bendi. As in the inaugural session and today's morning session, the scientists like Samvel sir and Srinivasan sir insisted on one topic, that is lab to field and the application of our findings to the downtrodden, that is the target people farmers. For that purpose, this field level demonstration is highly essential. So this was conducted through KVK. Actually, presently I'm working at Hassendare Perikulam, but it was conducted in Salem district, that is through KVK Sandiv. So next, please. See, as we all know, Bendi is the very nutritional crop for overcoming and to sort out the malnutrition and all. And the farmers mostly in the districts like Theni, Madurai and Salem district, they are cultivating vastly. But if you see this crop, although it has a lot of our varieties, co-varieties are there, they are preferring hybrid varieties for high yield. But the yield for the high, even if you all the breeders, if you concern, they are concentrating on the yield rather than the pest and disease resistance. So we have chosen and this topic based on our field survey. So wherever they cultivate, the farmers cultivate this hybrid varieties, we are facing, that is farmers are facing lot of pest and disease problems, and here they causes more than 40% loss. So here they are losing monetary benefit also. Because of lot of factors. One is the identification of pest and diseases. What is happening in the field? They are not knowing, their lack of knowledge on the identification of this, differentiating the stress, nutrition and all. And so what they are doing, as Shamil sir said his experience, I have also faced. So in the Salem district also, simply they will take the sample and they will be going to the input dealers and they will get the recommendation. So in that case, they are dumping insecticides in combinations. So we have a list of banned chemicals. What are all the banned chemicals? We are saying it is available in the market, and without knowledge, they are going for not only for banned insecticides, in combinations they are going. For example, if enloying is there, they are going for COC application also. So in that case, we thought of going into this field level demonstration, considering our IPDM, that is TNAU modules we have taken, and we have educated the farmers first with the objectives to pool all our IPDM components with what we have find through our university. And besides inputting all those through the demonstrations, earlier what we have did is we imported training for them in identification of the pest and how to install the trap. That is, even they don't know how to install the LOCK trap, pheromone traps and all. So we have trained them through our field and through our trainings at KVK. And we have identified, as we all know, 10 farmers in that particular bendy growing belts, and we have procured inputs, and we have distributed them to impart knowledge through them. Then that is through these 10 people, it will be reaching hundreds, it will be reaching thousands. In that way, we have planned this objective. Next, please. So in the Salem district, we have spotted the bendy growing regions, that is Panamarathapetti, Omalur, Vaikundam, Edapadi, and Mangalam, Puttur, and Yermanaikamatti, Egapuram. There also we have chosen the farmers who are interested in doing this IPDM practices, and we have chosen the plots, one at the distance of, that is farmers practices is taken into consideration, and our IPDM plot, that is field trial, and this common practice, farmers practice, we have chosen at the distance of about half a kilometer, 400 to 500 meters difference we have chosen, but that is the two within the same village. Then only we can take the farmers, they can comparison it. So within the field itself, we have chosen these two fields, and we have imported our technology intervention through, that is our TNAU practice, and it was conducted during 21-22, and here we have applied neem chem. So here when we consider this bendy, we were unable to concentrate on all the pests. So major pests were considered, mainly whiteflies and then borers. Although mite incidence was there, it was not accountable for record of the comparison, that is for along with the farmers practice. So we didn't taken that into account, and including instead we have come across a lot of sucking pests like whiteflies, and then here regarding the diseases, we have taken Cercospora wilt, which was because these regions, it was prone to those diseases, and when we considered this powdery mildew, it was very meager for recording. So we have concentrated on the major pest and diseases which were prevailing in that zone, and we have imported these packages. There is a soil application of neem cake, and trichoderma egg parasiticide for our fruit borer, and then seed treatment for disease, that is wilt, LOCK trap and pheromone trap for monitoring the sucking pest and moths, and removal of that is vein clearing disease, which is coming under our cultural practice, application of neem cake, and need-based only we have recommended, and in some cases, not in all the 10 farm fields, in some cases where the incidence, because of lot of reasons, that is they'll be practicing other agronomical practices, which includes excess application of nitrogen and all, that in those areas we come across lot of whiteflies incidents that and all. So in that cases, we have applied the need-based application of imidacloprid and spinosad for the management, and COC for the management of disease. And we have compared the farmers practice. As I told in the introduction itself, for the farmers practice, they were dumping insecticides after flowering within a week, at the weekly intervals, they will go for application of these insecticides. Not insecticides means it includes both insecticides, fungicides, growth regulators, some organics, everything.
I'm coming directly to the result.
Oh, okay. So this is our observation, that is in the demo plot, that is TNAU model IPDM plot. We come across less incidence of fruit damage, whiteflies, and then incidence of virus, wilt, as well as the leaf spot, that is Cercospora leaf spot. But in the farmers' practice, although it was found to be less, compared to our demonstration plot, it was found to be high. Next. And this incidence of this pest and diseases has a lot of impact on the yield. And as the yield shows, in the demonstration plot, it was high compared to the check, and which has lot of impact on the BC residue. That is where the benefit what they got is almost this is 2 rupees means that is 1.4. And here, if you see the gross the cost and all, it is comparatively increase. We can say the demonstration plot here we incur cost on the traps, especially traps and intakes. And the traps, since it is an initial stage, it was high. Rather, in the further what they have followed, it was found to be reduced also, which again increased the benefit ratio. Next, please. So this FLD, that is a field level demonstration, is highly essential. Only this is just a mere demonstration of among the 10 farmers is not essential. Further, this was taken through to the other farmers through our farm advisory, through diagnosis, field diagnosis, and our farm advisory, and all the seminars, that is farmer seminars and trainings what we have conducted in the KVK. And it has a lot of impact, because the farmers, when we go for impact study, they fetch fetch more price at the farm gate itself, because when they come across many consumers can come across the residue free commodities from these farms, they from the farm gate itself they fetch a lot of lot of marketing with high fetch of fetch price, that is marketing price, because since they have less risk. Because but this is earlier stage they were found to be, it was found to be tough for them, but when they got a good price at the farm gate itself, they tend to practice the IPDM practice. This is one of the success story from our KVK Santur. Thank you.
Thank you, ma'am. Due to the time shortage, we are not taking up any questions. Thank you. To all the speakers presenting here, who are going to present next, please stick on your time. It is 5 minutes, because time is very shortage to complete this session. So I request all you to directly come to your methodology and result sections rather than stick on to the introduction. So next speaker will be Dr. Chandramani, and Madam will be presenting on the topic assessment of organic silicon sources against tomato pinworm and its impact on the quality parameters of tomato. Okay, I will say. Okay. Sorry, it may be. Okay, sorry, it may be a confusion. So Madam will be delivering on the topic exploring silicon-induced volatile signaling and biophysical changes in green gram for defense against Mercovitata. Ma'am, over to you.
Good afternoon to all of you. I'm here to present exploring silicon-induced volatile signaling and biophysical changes in green gram for defense against Maruca vittata. We all know that green gram, Vigna radiata. Next slide. Mung bean is a poor man's meat. Among the vegetarians, it is grown in arid and semi-arid regions of the country. And the main constraint for poor yield in green gram is due to the attack of wide array of insect pests from seedling to maturity stage. So for managing the insect pests, in general, farmers are adapting insecticides. So in order to avoid 3 capital R problems, as Dr. Suganti pointed out, 3 capital R's, that is residue, resistance, and resurgence. So in order to avoid the 3 capital R problems, we are in a position to switch over to eco-friendly approaches. So one of the eco-friendly approaches is application of exogenous application of silicon. Exogenous application of silicon is highly essential because silicon depletion is noticed wherever monocropping is practiced. So when we apply silicon, it accumulates in plant cell walls, and which gives protection to the plant by enhancing the production of lignin and phenolic compounds. And in brief, it activates chemical defenses, particularly the synthesis of volatiles and non-volatile compounds. And also it enhances the physical structures like trichomes. Another important thing is by the addition of silicon, it attracts the natural enemies that use volatile cues to locate the prey. So with this background, the research was carried out with the two objectives. That is, foliar application of silicon was evaluated in combination with other eco-friendly approaches for pot borer, Maruca vitrata, and also biophysical features and volatile compounds as induced by silicon were analyzed. Next. This is about methodology. Field experiments. First field experiment was conducted at Agriculture College and Research Institute, Madurai. Another confirmatory trial at Farmers Field at Notabatti, Madurai district in RBD. And the variety chosen was VBN3. And two forms of silicon, silicic acid and potassium silicate were evaluated along with gibberellic acid through foliar application. The incidence of Maruca vitrata was recorded in 5 randomly selected plants, and biophysical traits, particularly silicified cells and trichomes, were analyzed as per the standardized procedures. And volatiles from herbivore damaged and undamaged leaves were also analyzed as per the standardized procedure. Additionally, orientation studies were also carried out using Chrysoperla. Next. And this is the result of the study. That is, among the 12 treatments, the mean number of Maruca vitrata larvae per plant was significantly less in the treatment with potassium silicate 0.5% and gibberellic acid 50 ppm, that is T7. Similar with the percent reduction of 54.87%, and the number of silicified cells was found to be highly significant in the treatment, and the number of trichomes were also found to be significant in the potassium silicate 1% and gibberellic acid 100 ppm, followed by, but which was on par with potassium silicate and gibberellic acid 50 ppm. Next. So this present study is in line with the agreement, in line with the findings of Nagaratna and co-workers 2022, who evaluated different forms of silicic acid, gibberellic acid and jasmonic acid, and they reported that the plants treated with silicic acid along with gibberellic acid exhibited lower larval pre-pupal and pupal weight. Indumadi 2020 evaluated the application of calcium silicate in sugarcane, and she reported more number of trichomes and silicified cells. And we have also calculated the correlation coefficient and the R value between the biophysical factors, silicified cells, trichome density, and pot borer incidence. And it was found to be, the R value was found to be significant and negatively correlated with the incidence of pot borer. That is, biophysical factors and the pot borer incidence were negatively correlated. Regarding volatiles, 8 plant volatile organic compounds were identified with more peak area in silicon supplemented damaged plant when compared to silicon supplemented healthy plants. And these compounds increase the attraction of natural enemy. Among these volatiles, ecosine was dominated in the volatile profile of damaged plant, which activates jasmonic acid pathway, and this compound has ovipositional deterrent activity. Koenig and co-workers 2011 reported the application of potassium silicate led to enhancement of N-heptadecane, making the plant more attractive to predatory beetle. Next. This is just to indicate the olfactometer study. This orientation response of Chrysoperla. The orientation response of Chrysoperla was significantly high after 1 hour, 2 hours, 3 hours, and 4 hours in silicon supplemented damaged plant when compared to silicon supplemented healthy plant. Next. Hence, in the present study, it is clearly understood that combination of silicon and plant growth regulators enhance certain biophysical traits, namely silicified cells and trichome density, which in turn resulted in induced resistance, which in turn resulted in reduced incidence of pot borer, particularly Maruca vitrata, thus imported induced resistance, particularly systemic acquired resistance. And the first picture is silicified cells, that indicates silicified cells. Next is trichomes. Thank you.
Thank you, ma'am. Now I call upon Dr. Jayarani, ma'am. Next, I call upon Gayatri, ma'am.
Good evening. I'm Dr. Gayatri, presently working as Assistant Professor in KVK Kattupakam, comes under Tamil Nadu Veterinary and Animal Sciences University. Today, my topic for presentation is adoption of pest management practices in brinjal, a case study in Chengalpet district farmers. Next slide, please. Actually, the brinjal in Chengalpet district, it is one of the most important vegetable which is grown in the area of 374.87 hectares with a productivity of 40 tons per hectare. And during our field visit, we have noticed that major portion of brinjal is affected by insects like borers, jassids, mealybugs, and mite, which is causing nearly about 40 to 80% of damages in the crop, resulting in yield losses. And most of the farmers were not practicing the very good management practices, particularly for insects. And our major objective of our study was to study the adoption pattern of the pest management practices which are being followed by the farmers in the district after KVK intervention. And to create awareness among the judicious use of the pesticides, and also to create the awareness among the farmers in identification of pests and diseases, which is one of the important factor which need to be emphasized in the farmers, because they are using the more amount of pesticides in the field, which is causing them economically, which is very cost effective, and also it is resulting in the poor quality of the produce. Next slide, please. The study was carried out in the 5 major villages in the Chengalpet districts, in Timmavaram, Hanumantapuram, Uttivakam, Nallattur, and Vadagal villages. And our interventions we carried out during the 3 years, that is 21, 22 to 23, 24. And these were the programs which we used for our intervention, that is training programs, YFTs, FLDs, and advisories, and along with diagnostic field visits. Nearly about 130 farmers were benefited because of these training programs, and major interventions which we made them to, or we recommended were the IPA modules like deep summer plowing, application of effective menus, that is mostly organic menus, then soil and seed treatment methods, balanced use of fertilizers, installation of traps, that is light traps and pheromone traps, and also yellow sticky traps, and border crops, and need-based pesticide application, also need-based biopesticides, and neem products. And data was collected in in-person mode, and basic statistical analysis was done for all our studies. Next slide. And in our results and discussion, the major pest, that is borers, were followed by mites, jassids, whiteflies, and mealybugs. These were the major pests in the brinjal growing areas. And the farmers, before our KVK intervention, they were not able to identify mites or the damage caused by the jassids. Based on our intervention, they were able to identify mite damage specifically, and also they were able to identify the little leaf of brinjal. Usually they were thinking that little leaf of brinjal is somewhat some God-given gift, and they were not uprooting the whole plants. After our interventions, they were able to identify and they were able to uproot the whole plant, and even they adopted the management practice like complete burning of the plants. Before that, they didn't use that, and they were thinking it is an ornamental plant, and just left like that. And soil application of bioagents was also done based on our intervention, but this soil application of bioagents were nearly followed by only 2% of the farmers. And results showed that majority of the farm, that is 68% of the farmers were able to identify pests and diseases after our KVK interventions. And application of the farmer menus were nearly about 92% of the farmers were following the application of farmer menus. Next slide, please. And based on the KVK interventions, 35% of farmers adopted the clipping of the shoot and that is borer affected plants, because they followed this practice. They were easily adapted to this practice because it was very cost effective, and it was also very easy for them to follow this practice. And most of the farmers in our district, they were following only the pesticides which were recommended by the input dealers. That is 76% of the farmers were following the pesticide application based on their input dealers' recommendations. And effectiveness of pheromone traps and light traps, it was only about 3%, because the availability of pheromone traps and lures was not available in the local market. Only through KVK it was made available. Otherwise, it was not available in the local market. So this only 3% of the farmers were following this type of recommendation. And compared to KVK intervention and the farmers' method of practice, nearly about 9% of yield increase was noticed, and the yield was nearly about 320 quintals per hectare yield was obtained, which was very satisfactory for the farmer side. Next slide, please. These are some of the action photos of our interventions. Next slide. And when you see the salient findings or conclusion, nearly about 90% of yield increase was recorded in the farmers' field, and farmers were able to identify the pest symptoms, and they adapted accordingly the management practices. Before that, they were as such, means just like that, they used to spray chemicals. After KVK interventions, nearly about 50% of farmers, they were able to follow need-based chemical pesticide application. And many success stories of these farmers are recorded by DD Podige and All India Radio. And the major constraint in adopting this IPM practice was mainly the availability of pheromone traps, lures, and reliable biopesticides. And thank you. Any
Thank you, ma'am.
questions? Okay, thank you. Since because in Extension Act side, we used only this type of data. Thank you.
Thank you, ma'am. Now I call upon Dr. Sujay Anand.
Respected Chair, Co-Chair, distinguished participants, a pleasant good evening to all. Today I am going to present my topic on the biointensive IPM module for chickpea, enhancing the crop health and sustainable productivity. So, excuse me. Thank you. So we all know chickpea is one of the major pulse crop. It is the primary, most important pulse crop cultivated in India, and the major pulse, all other pulses comes after chickpea, like pigeon pea, then Vigna crops. And the total hectares covered in India is 104 lakh hectares. And with the production of 122 lakh tonnes, chickpea stands first in India in area and production also. And when we see the productivity of India, it is only 1,261 kg per hectare, which is very low than the Australia, which is having around 1,725 kg per hectare in the global scenario. And moreover, it is one of the important export crop also. Among the pulses, chickpea overall countries, India is the second largest exporter of chickpea in this world, followed by Australia. And when we see the major importers of chickpea from India, it is Algeria, Turkey, UAE, followed by Pakistan and other Middle East countries. And further, chickpea is one of the major crop in the most of the culinary dishes in Mediterranean region and Southeast Asian region. Then, when we see overall India, 25 states cultivate chickpea, and the entire area of chickpea cultivation can be classified into 5 different zones. One is Northwest Plain Zone, Northeast Plain Zone, West Central Zone, East Central Zone, and South Zone. And the 95% of the chickpea production comes from the Madhya Pradesh, Rajasthan, Maharashtra, Gujarat, UP, Andhra Pradesh, Karnataka, Chhattisgarh, Bihar, and Jharkhand. And in all this region, when we see the major pests, that is gram pod borer, fusarium wilt, then root knot nematode. These 3 are the major constraints in increasing the chickpea productivity in India, in all the zones majorly. Hence, we had framed our objectives as identification of a suitable bio-intensive IPM module for managing these 3 major insect pests. And in the simultaneously, as we all know, pulse crop enriches the soil nutrient status. So it also should improve the environmental health. So we want to conserve the natural enemies present in that respective ecosystem. Keeping these 2 as the major objectives, we had proceeded the experiment with the following modules. So totally 7 different treatments are there. In that 5 are having the biointensive IPM modules, and 6th treatment is the farmer's practice, and 7th is the untreated control, that is check. 6 and 7 are sole crops, and in the intercrops we had taken lentil in the first BIPM module, coriander, fennel, fenugreek, and mustard. And the ratio of the main crop to intercrop has been given in brackets. In the basal application, we had applied a neem cake in all the 5 biointensive IPM modules, and we had followed a seed treatment of Dullanderma, the Trichoderma asperulum, our own IAPR product, at the rate of 10 gram per kg of seeds. And after the ETL has crossed, in case of pod borer, we had gone for spraying a BT liquid formulation known as IAPR Pulse Guard. In case of the farmer's practice, sole crop was taken, and seed treatment with tebuconazole and carbosulfan for this one. Then in case of pod borer, we had used flubendiamide. And the experiment was laid out in randomized block design with 3 replications, and the plot size is 5 by 5. And as per the recommended package of practices for the Northeast Plain zone, we had followed the agronomic practices without any insecticidal spray initially. We are recording the natural enemy and the pod borer incidence at the 5 randomly tagged plants. And we had calculated the Simpson's index and Simpson's index of diversity. The formulas were given. Just I highlight the utility of the Simpson's index of diversity. It gives the species evenness and richness of that module what we are using. Then Sorensen coefficient was also taken. Sorensen coefficient, we had compared the module with the farmer's practice. So what is the unique species present over than the farmer's practice, that will be indicated in the Sorensen coefficient. With this background, we had started the experiment. In the after 30 days of sowing, we had collected the rhizosporic soil samples, and we had examined the nematodes species present in that one. Then we had calculated the yield, and from that yield data, we had calculated the benefit cost ratio and the rate of returns of the investment. Then apart from this, a pot experiment was also conducted simultaneously with the chickpea and the intercrops and all the treatments, wherein we had released the 100 cc of nematodes, root knot nematode, Melanogaster javanica, and we had noted down the root galls in the each module to quantify it. And when we see the results, the coccinellid population and syrphid population were very highest in the BIPM2, that is chickpea with coriander. All the module photos has been given. And further, the pod borer infestation was also the lowest in the BIPM2 module. And percent pod damage was also 0.49. All these were statistically analyzed using the SPSS software, and the percent reduction over control was also calculated. Further, interestingly, we had noticed many unique syrphid species having 2 different genus. One is the Ichidon, and another is the Eristalis genus in the case of chickpea plus coriander intercropping. And we had noticed many dipteran parasitoids and pollinators in case of chickpea plus coriander system. That may be an insectivorous, nectarivorous plant, which may be the reason for having more insect diversity. Further, from the soil samples, what we had calculated, we had not found any significant difference in the presence of Tylenkids and Rhabdidids in the soil sample. Then Fusarium also, there was no significant difference from the soil samples what we had collected. In case of pot experiments, what we had done, we had noted the root galls per plant was lowest in the module number 5. That is having chickpea plus mustard as the intercrop.
Your time is over, sir. Please wind up.
Okay. Coming to the final conclusion, that the BIPM2 module has recorded the highest benefit cost ratio and the return of investment. Similarly, BIPM3 and also the highest insect diversity. Lowest Simpson diversity index was noted in the BIPM2 module. This was the take-home message, that the BIPM2 module is having highest coccinellid population and the syrphid population and lowest pod borer infestation. And similarly, it has simultaneously recorded the highest benefit cost ratio and the return on investment. Definitely, this BIPM module will help in enhancing the crop health of chickpea as well as the environment health. As already we had attained the self-sufficiency in the pulse production. So it is the time to think over for a healthy environment. So hence this project will be help in achieving the objective of this conference. So thank you one and all, and the organizing team for giving me this opportunity.
Thank you, Dr. Sujay. Now I call upon Dr. Vinod Kumar. Okay. The next presenter is Dr. Indira Gandhi.
Folder name OHPGPR. No, not available. No, that is not. Oh, okay, madam. Okay. Actually, I was sent to the committee. Yeah.
Shall I? Good afternoon. I'm Dr. B. Vinod Kumar. I'm going to present about management of major pests of strawberry through eco-friendly approaches in Nilgiri district. See, once upon a time, this strawberry is considered as fancy food. And after knowing the nutritive value of the strawberry, now the inland consumption of the strawberry is very high. Next. Very high. And major challenges in the strawberry production is the insect pest and diseases. Now it is cultivated in the protected cultivation, and here the sucking pest is dominating. And nowadays, to manage this pest and so many pests, it is occur in the strawberry fruits, and it leads to the food safety issues. So with this background, see, in the Nilgiri district is now it is considered as organic district, and it is declared as the organic district, and use of these pesticides is prohibited. So the use of non-chemical alternative is gaining importance now. So with this background, I have the study has been conducted to know the seasonal abundance of major pests of strawberry and to evaluate the different botanicals and microbial agents for the management of major pests of strawberry. So trial was conducted in the Pali House at HRS Vijayanagaram, Ooty, and the incidence of major pests of strawberry crop was recorded at fortnight interval from January 2023 to December 2024. And the weather parameter also recorded simultaneously, and this weather parameters is correlated with the pest incidence, and the weather-based forewarning model was developed. And 2 field studies were conducted to protect in the protected condition to evaluate the botanicals and microbials for the management of major pests of strawberry. See, for the botanicals, based on the earlier study, we have taken 3 plants. One is Ageratina adenophora and Ruta calapensis and Artemisia nilgerica. These 3 plants already it is in earlier studies for other crops. It is proven that insecticidal property was proven, and 3% crude aqueous solution was taken for this study. And microbials, Metarrhizium municipale and Bavaria bassiana and Lecanicilium lecani. These 3 are we are producing it in Nilgiris. And these 3 microbials we have taken for the evaluation study. And the standard check, 3G extract and 5-leaf extract, it is produced in the Department of the Organic Agriculture Department, from where we got this as a standard check, and uncontrolled unsprayed check also it is maintained for the comparison. The result shows that the seasonal incidence in the seasonal incidence study, the incidence of sucking pest is dominated in the strawberry rather than the defoliators and borers. And incidence of thrips and mites is high during the summer and started decreasing after the onset of monsoon. And incidence of aphids, it is high during the monsoon season, that is September to October. And during winter season, all the pests are generally it is very low. And here we have I have calculated and we have developed a weather-based forewarning model. So here the TRIPS is it is positively correlated with this. We have taken only 4 weather parameters. Since this study is conducted in the protected cultivation, only maximum temperature and minimum temperature is taken, and humidity in morning and evening humidity alone, RH alone, it is taken for the study, because the weather this is rainfall and wind velocity, everything is not having any impact in the polyhouse. So only 4 weather parameters is taken for the analysis. So here the thrips and mites, it is positively correlated with the maximum and minimum temperature and negatively correlated with the humidity. Whereas in the aphids, see here X1 and X2, X1 is the maximum temperature, X2 is the minimum temperature, and X3 is the morning relative humidity, and X4 is the evening relative humidity. Here the multiple regression equation model was run. Here the evening relative humidity is not having any impact on the pest incidence in the strawberry. So here the mites and thrips also, it is positively correlated with the temperature and negatively correlated with the humidity. And aphids, here it is negatively correlated with the evening temperature. Okay, sorry, minimum temperature. And Spodoptera, it is a defoliator, it is negatively correlated with the RH. Whereas in the ash weevil, it is a minor pest, even though it is only 73% of its incidence is, depends on the weather parameters, and it is negatively correlated with the minimum temperature. So here, the minimum temperature having the great role in the incidence of these pests. So evaluation study we have taken here. See, Ageratina adenophyra, it is crude extract 3%. It is having the great impact, and it is the reduce the population. It is the mean data for the 2 seasonal study and 2 seasonal study and 3 replications. Okay, here the thrips incidence, it's more than 81% of thrips was population was reduced, and Spodoptera population also, it is having the great impact on the management. It is 86% of pest reduction was observed in the Ageratina adenophyra crude extract. It is followed by Artemisia nigricans. It is having only little bit effect on the thrips. Whether the other pest, it is Rutacalapensis and Artemisia nigricans not shown any great impact. Whereas this 3G extract, it shown good results. It is 71% of mite population was reduced in the 3G extract, and 5-leaf extract, it is thrips population, it is almost 80% of the thrips population was reduced in the standard check, that is 5-leaf extract. In the Metarhizium, Bavarian, Licalinicillium, this Metarhizium and Bavarian, these true entomopathogenic fungus, it is having almost 62% of population of Spodoptera was reduced, and in the Bavaria bassiana, it is only 58%. It is the reduction of population was observed. And this Ageratina adenophyra, it is the crude extract, it is taken, and for the GCMS analysis, here we have identified 5 active compounds. So here dicocidinate, this is having the more than 5.24% bioactive compounds. It is present in the aqueous extract of this Ageratina adenophyra. Here it is bioactive against insects and disrupt the metabolism. Okay, yeah, these 5 molecules, it is having the potential for the further studies and for the development of new molecules. So here the salient findings or conclusion of this study is the Ageratina adenophyra aqueous extract, it is 3% can be added as the potential and eco-friendly component for the integrated pest management package in the strawberry. See here, this Ageratina adenophyra, it is, if the concentration is increases, it is act as the antefidant, and when the concentration is reduced or low, see it is showing the growth regulation effect. This is the moribund larva. The moribund larva here, see, this is a chitin synthesis inhibitor effect. It is showing in the by this plant. So it is this plant having the great role, and this plant may be taken for the further study. So here already it is taken for the formulation, and formulation study is going on, and soon it is given for the OFT and FLD. Thank you.
Thank you, Doctor. And one more, actually, this 5-leaf extract, the recommended concentration is 10%.
Is it so much? Okay.
Yeah. 3G is the 5% will be okay, but for 5-leaf, we need 10%.
Okay, madam.
Okay. Thank you.
Thank you.
Now I call upon Dr. Indira Gandhi. இருக்கடா கண்டுபிடிச்சிட்டீங்களா? PDF, first one.
Yes. Respected chairman, co-chairman, and the rapporteurs of this session, very good evening to all of you. Here I am going to present about the effect of different bio-intensive pest management modules against the major insect pest of sesame with reference to organic sesame cultivation. So, as usual, irrespective of horticulture and agriculture, all the crops are highly important and essential one for our human beings. So with reference to sesame, what is the speciality means? This is the well-known crop domesticated even 5,000 years ago. So it is known as queen of oil seeds because of the rich antioxidants present in it, and low input crop come up very well in the hungry and thirsty soils also. So the tiny seed with nutritional punch captivating the taste bud and market. However, the demerits of sesame is low harvest index, seed shattering, and lack of high yielding varieties and resistant varieties for abiotic and biotic resistance. With reference to our topic of the discussion and the conference, biotic stress, major biotic stress is the insect pest. In light of this, we have formulated different IPM modules, that is bio-intensive IPM modules, to evaluate against the different insect pest. Okay, each and every crop is infested by different insect pests. What are all those and which pest is causing major damage and leads to major yield loss means this is the list of insect pests causing damage to sesame crop. With reference to the damage intensity, I have arranged the pictures. Here you can see the pod bug. First one is the antigastra. This is causing damage in the 3 critical stages: vegetative, flowering, and capsule formation stage. Whereas the pod bug, this is the special one because it is causing damage under field and stored condition. Then the last one, the dialect, I can quote last but not the least, that is the leafhopper, major dreadful phyllodes disease transmitted by this leafhopper vector. So how to manage all these pests means we have to go for bio-intensive model. So I have told about that leafhopper species, they are causing and that is transmitting vectoring the phytoplasma disease. So far 2 leafhopper species are proven for their transmission ability. We have studied 3 more species. Here I have depicted the 3 more species. In addition to Voracious Hicimonas, Bulclutha, Thomsonia, and Extinias are the 3 more species. But transmission ability of these 3 leafhoppers under Tamil Nadu condition is yet to be explored. So with respect to other insect pest, this leafhopper is causing phyllodes. So this is the different phyllodes expression in a single season, right from the vegetative stage to till harvesting. In each and every stage of the crop is facing and expressing the different phyllodes symptoms. Here we can see the floral sterility also. So because of this phyllodes, normally we used to say the term disease, but originally transmitted by vector. So this phyllodes disease vectored by leafhopper causing even up to 100% yield loss. Okay, this is the background of this experiment. So in light of this, we have formulated that objective. So for that, we have worked for 3 years. Actually, here I have mentioned only 1 year, sir. We have conducted this experiment for during Kharif 22, 23, and 24. So we have taken up this experiment with the popular sesame variety VR82. The experiment was conducted at a new farm, the regional research station Virudhachalam. Actually, regional research station Virudhachalam is one of the lead center for oilseed research, including groundnut and sesame. Okay, so randomized black design was formulated. The each and every treatment was replicated thrice. After layout and sowing taken up, we have observed the sesame, leaf webber, and capsule borer on 7th, 14th day after treatment imposed and damage percent and leafhopper population, natural enemies ultimately yield under BC ratio. So here we can see the treatment difference for audience quick capturing. 1, 3 and 5 are having some similarity. The similarity are application of farmyard manure followed by neem cake. The first one is common. Then second one is if the farmyard manure is the basal one, means there we have applied trichoderma, basal and enriched trichoderma viride. In the case of 2, 4, 6, this treatment, application of vermicompost, then followed by Pseudomonas fluorescens. As usual, basal and enriched with farmyard manure applied as foliar one. Then in addition to these two parameters, we have sprayed that neem seed kernel extract, neem oil and Bacillus thuringiensis variety Krustaki. This is the difference with respect to 1, 3, 5 treatment. Then in addition to these, we have raised maize as a border crop and also released that is Trichogramma chylonis egg parasitoid. These are all the treatment details. Because of these treatment imposement, what we have observed was the treatment T was found to be an effective one in reducing the antigastra damage and reducing the leafhopper population. Ultimately, the phyllodes incidence right from flowering to reproductive stage. In addition, as usual, our aim is to reduce the pesticide 3R problems and improve the soil health, plant health, ultimately human health. So the natural enemies population also more in 45 days. Here, the ultimate aim is, of course, high yield. So when compared to other treatments, T3 found to be an effective one, which was recorded 590 kg of sesame seeds per hectare and recorded the highest cost benefit ratio. So salient finding is, as I said earlier, T3 was found to be an effective BIPM module for sesame pest management. So the organic sesame production or natural farming relies on cultural practices, natural enemies, natural fertilizer and biological control agents with the aim of improving the soil productivity, health and supporting the plant nutrients and ultimately the controlling not only insect pests but also weeds and other biotic stresses. So the effective BIPM module would serve the purpose in organic or natural farming system of crop production with a special reference to sesame by containing the insect pest population and improving the health of right from the soil to plant, animal, finally the human being. Thank you very much for your interest hearing.
Thank you.
Actually, by thank you slide, what I want to say is from the pest infected sesame, because of our biointensive module, the sesame feeling happy. So that is what I want to insist to the house. Thank you very much.
Thank you. Thank you, Doctor. And now I call upon Dr. C. Shankar, sir. Now I call Dr. Jayarani, ma'am.
Good evening to one and all present here. Myself, Jayarani, entomology professor working in TNAU. And now I am going to briefly present a small work which was undertaken at Kurmiyan Malai when I was there. So actually this is something which we felt, that is when we saw, we are talking about rhinoceros beetle and their grubs. So, we have some few palms, coconut palms at our farm. So where we have a problem of fronds, which is drying and falling down, and it is getting accumulated, and the waste disposal, we encountered a problem. So from that, I have taken this idea. So we know the rhinoceros beetle, it is feeding on the fronds, and it could able to digest the cellulose material which is present in the frond. So we would like to ensure whether this young stages of the rhinoceros beetle is harboring any symbionts that could have the ability to degrade these things. So with that idea, we took this small study and we have collected the rhinoceros grub from two areas. One is from the Coimbatore and another one from the Kudumiyanmalai itself. Next. So we, because in our sharing session, we excluded this introduction, but since this idea where from we got, we wanted to express, that's why I have included the introduction. Sorry for that. Okay, so we have framed our objective to identify, isolate and identify the gut symbionts from the rhinoceros beetle grub, which is easy rather than dissecting a adult, where it is rhinoceros beetle. Okay, and then another thing is to evaluate their effectiveness in decomposing the materials, waste materials, that is agricultural waste, which is a problematic thing nowadays. And with this objective, we have collected the rhinoceros grub from the two locations. One is from Coimbatore and another one is from the Kudumiyanmalai campus. And we have isolated and identified the bacterial symbionts, which is present in that. And then that bacterial symbionts were evaluated for their efficacy in harnessing, enhancing the decomposing process. Next. So from the isolation, we could able to isolate 6 isolates, bacterial isolates from the Kudumiyanmalai area and 4 from the Coimbatore district, and most of which, based on the morphological thing, we have identified up to the genus level. Since I told it is a brief work, so we have identified based on the morphological characters as bacillus species mostly present. So in Coimbatore, we have 4 isolates, of which 2 are bacillus species only, but they have different morphology. And the contribution or the ratio in the plating is, one is 76%, that is bacillus species B3 was occupying 76% of the content, gut content, and the another one is 0.96%, that is named as B4, that is bacillus 4. From Kudumiyanmalai, we have isolated methanobacter species, bacillus species B1 and Pseudomonas species. Actually, to our surprise, we could able to isolate a Pseudomonas fluorescens, clear-cut Pseudomonas fluorescens, because nowadays we are having the conflicts, whether it is really Pseudomonas fluorescens or it is Pseudomonas aeruginosa, which is a human pathogen. But to our surprise, we could able to get Pseudomonas fluorescens from the Kudumiyanmalai isolate, and that was the species name was confirmed with the Dean Nakiran sir. And the another one is bacillus species B2. Here actually the ratio is somewhat very low in the Kudumiyanmalai campus. And then after this isolation, next slide. We have made 20 different treatment combination. That is, one is, so for normal composting, we'll be using the waste material along with the cow dung for composting. And so we took the normal method of composting, and another one is without any cow dung addition. And so these 2 treatments were included along with our isolates. So all the isolates except the Pseudomonas fluorescens, we didn't include that in the treatment schedule. Okay, and when it was applied on the composting material, and this was the isolates were sprayed once in 15 days. At least it is 3 times, because the composting period almost 60 days. So starting from the first day, it was sprayed 3 times, and we could able to analyze the carbon and nitrogen content of the composted material in the end of the process. And we could found that the isolate from the Kudumiyanmalai, the bacillus species could able to enhance the composting process even before the 60 days. And with the carbon nitrogen ratio was 20:1. So strictly speaking, the best preferred carbon nitrogen ratio for a compost should be 30:1. And here it is 20:1, which is almost closer, and with the shorter time, that is 45 to 50 days, we could able to compost this. So this was proved to be an efficient isolate. So next slide. So to conclude my topic, so there is a possibility if you isolate the gut symbionts from different area, different locality of the grubs, rhinoceros grubs, not only grubs, and also the further research has to be extended to the rhinoceros beetle, because rhinoceros beetle is nowadays alarming or alerting status of pest, and there is a recent report that is stating that it is also feeding on the corn cob. So we can collect, and that is another mechanical way of controlling that pest also. So we can explore the gut symbionts of that beetle adults as well as the grubs, and so that we could arrive at definitely a solution for composting the waste. So we can avoid the pollution because of this waste accumulation in the ensuing period. So this, it's a basic, and this will be further worked into a thesis research work. Thank you.
Thank you, ma'am. Any questions?
Thank
you,
you.
ma'am. Now I call Dr. Shankar.
First of all. Good evening to all. My name is Dr. Shankar, KVK, Namakkal, Tanuvas. Now I am going to present the impact of IPM strategies for effective management of major insect pest in watermelon. Before that, watermelons everybody is eating, but whether it is organic or inorganic, then how much pesticide is there, we don't know. So in that case, during my PhD, I did this work. Before that, I want to introduce about watermelon. Watermelons have more vitamins and minerals, and also that it is production in India. It is third place. China is the first place, and also in Tamil Nadu, various districts it is continuously cultivating these crops, and also farmers are continuously cultivating. Farmers are using indiscriminate use of pesticides and also overdose application of the pesticides. So it causes lead the severe damage to the crops, and also losses, and also development of resistance also is there. So in that case, this I have. In that case, I took the objective of to development of IPM strategy for effective management of major insect pest in watermelon. So the trial was conducted at Namakkal district in farmer field with different components of the treatments. That common is one is the eco-fish crops, then botanicals plus biorational pesticides, then application of need-based chemical pesticides. I have taken the 6 treatments and conducted in farmer field, and also I took observation 15 days intervals after the pre-post spray, then after post spray. And also I have observed the sucking pest, leaf folder, and fruit borer incidency. Among the treatment, the first one is that cluster bean plus neem oil plus biorational pesticide Bavaria, then need-based application of that chlorotoluene is pest method for reduction of the pest in watermelon crop. And also we have I have analyzed the residue level also. It is below level of the residues was there in this trial crop. Now the trial is supported the different scientists in my research. This is the graphical reduction of the different insect pests in watermelon. Then regarding the conclusion, already I told that the first practice treatment is that cluster bean, then neem oil, then Bavaria, then based on need application of coragen was effective reduction of the pest incidence in watermelon, and also increasing the yield. This I thank for the opportunity to give the research in TNAU as well as Madurai and Tanuvas. Thank you, sir.
Thank you, sir, for keeping up the time. Thank you. So any questions? So if there are no questions, we'll end up the session. Pranila?
That concludes our oral presentations. We extend our gratitude to all the speakers for their valuable contributions. As a symbol of our deepest gratitude, we would like to honor the panelists. I request Dr. Guru Prasanna Pandey sir to honor Dr. M. Shuganti ma'am. I request Dr. M. Shuganti ma'am honor Dr. Guru Prasanna Pandey sir. Convener Dr. G.K. Sujayanand sir, Senior Scientist, IAPR Kanpur. Rapporteur 1, Dr. S. Suganya Kanna ma'am, Associate Professor, Entomology. Thank you, everyone. Tea has been arranged and it is placed outside. You can have it.